My project is on CRISPR and if it's possible to remove disease from DNA using this mechanism. Do you have hitchhiker's thumb? Can you roll your tongue? Do you have a widow's peak? These are all answers that are encoded in your DNA along with other instructions that gives you certain traits, skills, and susceptibilities. Your DNA also contains information on your health, both your current status and your chances of developing certain diseases. Genetic testing can help tell you whether you have certain genes related to higher odds of having diseases. In some cases, that knowledge can help you make decisions about prevention. But what good is knowing your chances of having diseases such as macular degeneration, Huntington's, Parkinson's, or Alzheimer's if you can't prevent them? What if I told you that there is a procedure that you could rewrite your DNA that says you are likely to develop a particular disease? Or maybe rewrite the DNA that caused the disease you're living with right now. This is what scientists all around the world are doing in their labs. With the discovery of a new gene editing technique called CRISPR-Cas9 or CRISPR, scientists can now change the DNA of animals and plants and even human cells in petri dishes. Now, in early clinical trials in humans in the United States and abroad, CRISPR could one day offer a cure for a host of diseases. So, how does CRISPR work? Almost every cell in our body has a genome. Our genome is our complete set of DNA that makes us unique. To compare, it is like a personal barcode. To edit DNA, scientists use a process they observed in E. coli bacteria. E. coli has a built-in immune system that enables it to destroy viruses that may try to attack it. CRISPR refers to the DNA sequences in the bacteria's genome that will help it identify threatening viruses. When the system is triggered, the bacteria cut the DNA of the offending virus in half. Scientists are now learning to use that same DNA cutting process to cut sections of DNA in plant, animal, and human cells to edit away parts that could cause diseases or other problems. After the DNA is cut, the cell realizes its DNA is damaged and tries to repair it. Scientists can use the cell's built-in DNA repair machinery to then introduce the changes they want to make to that genome. This type of gene editing, like using word processing software to delete or correct a typo, can shut off a gene or change the order of its genome letters. With a single treatment, researchers recently cured a rare liver disorder in mice by editing the mutated gene that caused the disease. CRISPR might also help the researchers make more specific antibiotics that can kill disease-causing bacteria without eliminating good bacteria. If successful, CRISPR could one day edit away virtually any illness driven by a genetic mutation, be it in the DNA in your own cells or in the cells of a virus that has invaded your body. The BRCA1 gene or BRCA1 gene that substantially increases risk for breast cancer, the genes that cause cystic fibrosis, macular dystrophy, their genetic codes found in your genome inside almost every cell in your body could be snipped out. Researchers are trying out CRISPR in many diseases. The first attempt to use the technique in humans began last October in clinical trial in China. Scientists injected CRISPR cells intended to destroy non-small cell lung cancer into a patient with an aggressive form of the disease. They don't have results yet. The study is still recruiting participants and will be complete in April 2018. In the first CRISPR trial, to get green light in the U.S., doctors at the University of Pennsylvania will test the safety of CRISPR therapy in 18 people with various cancers. The trial has not yet started. Elsewhere, scientists are searching for a CRISPR-based cure for HIV. Attempts so far have failed. Still, scientists are pressing on. Cancer and HIV are just the tip of the iceberg. So, what are the risks and benefits? CRISPR isn't science's first method of gene editing, but it is the easiest, most precise, and most broadly applicable. The speed and ease with which CRISPR allows researchers to edit genes puts the pressure on bioethicists and regulators to address a bevy of concerns about the technology in a hurry. It would be difficult to predict the consequences of that inadvertent editing. Researchers at several institutions are exploring techniques to ensure that CRISPR cells edit only their specific targets. There are some ethical concerns. Technical glitches aside, CRISPR gives humans a power to manipulate life that they have never had before. Whether it's for use in people, crops, or entire insect populations, CRISPR raises countless ethical questions. While the researchers who developed this method of gene editing are interested in advancing science and potentially curing human disease, The same technology would theoretically allow specialists to edit DNA in eggs and sperm and create designer babies in vitro. There may be some things in an egg or sperm's DNA that would be unethical not to change. If you can turn a lethal disease, or even worse, a disease that does not claim life quickly but rather leads to a life of misery, would it not be good to do something? But editing the genes of eggs and sperm changes the DNA that would be passed down to offspring for all generations to come. Some argue that humans should not have this level of control over human reproduction under any circumstances. Even those who would support editing DNA in the name of disease prevention might ask where to draw the line. The National Academy of Sciences and National Academy of Medicine acknowledged this question in a recent report. The academies support continued research into gene editing aimed at treating and preventing diseases in cells that are not passed on to offspring. They support research into editing inherited genes only in the case of serious diseases and disabilities. The report admonished scientists to hold off on gene editing intended to enhance an otherwise healthy genome. That is, don't create designer babies just yet, a concept that's still very far away from becoming a realistic possibility. Given the potential benefits, CRISPR-based cancer treatments might be among the least controversial and perhaps the first that doctors may be able to offer their patients.